It's my pleasure to begin our afternoon session by introducing our second next-generation leader, Tom Nowakowski. Tom is an assistant professor in the departments of anatomy and psychiatry at the University of California at San Francisco. During his doctoral studies with David Price at the University of Edinburgh, he interrogated the role of microRNAs in radial glial maturation and glial neurogenesis. As a postdoctoral fellow with Arnold Krigstein at UCSF, Tom used single-cell mRNA sequencing to characterize cellular diversity in the developing brain. And his new lab investigates the development of the human cerebral cortex using high-throughput single-cell genomics approaches. He will give a talk today entitled Development and Area Formation in the Cerebral Cortex. Please welcome Tom Nowakowski.
Great, thank you so much for the introduction, and it's a real pleasure to be here. And I'm going to talk about the human cerebral cortex, which many of you know quite a lot about. It's a structure that's sort of most complex in the brain, and many people believe that it's responsible for many of the sort of higher order cognitive functions. It's probably the organ that you use to decide to come here today and learn more about the brain, so it's a really interesting structure. And the cellular complexity of the cells that exist in the cerebral cortex has been appreciated since over 100 years ago, since the days of Santiago Ramon y Cajal. It's been appreciated since then, it's been estimated since then, that human cerebral cortex contains billions of neurons, excitatory neurons distributed across dozens of functional areas. And when you look and compare neuroanatomy across different areas, you will learn to appreciate that there's quite a lot of variation in terms of the cellular compositions and distribution, including laminar differences across distinct anatomical areas that have been appreciated since the days of von Economo and Brodmann. And yet, these differences emerged during early development from an apparently uniform neuroepithelium that has been described in studies, again, since the days of early anatomists, since the days of Wilhelm His, who illustrated some of the limited diversity of cell types that exist there. And central to the formation or the generation of the diverse excitatory neurons in the human cortex are a population of cells that are found only transiently during development that are called radial glia cells. And these cells possess these long fibers that extend all the way to the peel surface from the ventricular surface, where these cells undergo mitotic divisions to generate postmitotic neurons. And the neurons that are generated from radial glia then utilize the radial fiber to migrate to the terminal positions in the cortical plate. And upon their deposition in the cortical plate, they actually arrange themselves into those structures or arrangements known as the mini-columns in the cerebral cortex. And there's quite a lot of interest in trying to understand the function of these mini-columns. We know that the diversity of cells or their laminar position in the cerebral cortex during development is determined by the birth date of neurons, since this studies performed by Pashka Rakic in the 70s, especially the macaque. But since then, we've learned quite a lot about the molecular diversity of these radial glia cells and their arrangements and developmental histories. And so we often think of the radial glia cells as generating or contributing a unitary production of excitatory neurons to the cortical plate, and that these cells are then arranged in a stereotypic fashion that is largely similar across different cortical areas. Thanks to many of the genetic studies performed mostly in mouse, we've identified some of the markers that allow us to visualize these radial glia cells, and then also transient amplifying populations of intermediate progenitor cells that amplify the number of neurons that are generated during development. When you compare that situation with the human cortex, however, you see quite a lot of differences. And in particular, earlier stages of development are very, very similar to those in a mouse. So shown here in red are radial glia cells, shown in green are intermediate progenitor cells. And early in development, these two pictures look very similar. But when you look at later stages of human development, and that's also true in macaque and also in other species, you see this major expansion of the radial glia and intermediate progenitor cell population in the outer subventricular zone, an anatomically distinct region that does not form in the developing mouse. And the radial glia cells that are present in that compartment possess cell bodies that exist in the, occupy the outer subventricular zone, and they still possess this long fiber that supports radial migration to the cortical plate, but they lack direct contact with the ventricular surface. But until recently, we didn't really have molecular markers to be able to selectively visualize these cell types. And so together with another postdoc in the Kriegstein lab, Alex Pollan, we performed single-cell RNA sequencing, a technology that allows you to identify gene expression profiles in individual cells, where we microdissected primary cells from the developing cortex, from the ventricular zone and outer subventricular zone, and using unbiased methods, we identified major sources of transcriptional variation that allowed us to pinpoint these radial glia cells. And later, by iterative clustering, we identify a transcriptional source of variation that highlighted a distinction as to whether these cells were sampled from the outer subventricular zone or from the ventricular zone, which then pinpointed several of the gene candidates that we were able to later validate by staining for them in histological sections with these markers visualizing or highlighted over here, labeling outer radial glia cells, and these genes labeling ventricular radial glia cells. So what started off as a uniform population could now be distinguished by selectively labeling two distinct populations in the ventricular surface and in the outer subventricular zone, and very similar findings that were actually reported by a parallel publication from the Allen Institute, Thompson et al., which also pinpointed some of the same genes. With these markers, one of the utility that you have is that now you can survey the abundance of these molecularly distinct cell types across developmental time points. And that's exactly what we did. So we stained primary sections of the developing brain with these markers, and to our surprise, the transcriptional state that we attributed to the ventricular radial glia cells turned out to actually emerge very sharply during development. That was quite surprising. The other thing that was very surprising was that the outer subventricular zone markers, including this HOPX gene, was actually labeling many of the, or vast majority of the radial fibers that were arriving at the cortical plate past about mid-gestation. And that was also quite surprising, because it suggested that these outer radial glia cells at some point in development may support radial migration to the cortical plate solely. So when you look at the comparable image in the ventricular zone, you will see that these cryo-B positive ventricular radial glia cells have fibers, but most of these fibers terminate in the outer subventricular zone. Many of these fibers tend to run tangentially, suggesting that they may not actually reach the PL surface, whereas the outer subventricular zone radial glia cells start in the OSVZ, and then they extend the fibers to the PL surface. Many of these morphologies have been identified in the past and described by Pashka Rakic and others, but the timing of their generation has been sort of elusive. And thanks to the molecular markers that we could now, that we've identified through single-cell RNA sequencing, we were able to determine the exact times in development when these cell types emerge. But this also highlighted an interesting possibility that at some point in the development, the radial glia scaffold that shapes our understanding of how developing cerebral cortex is organized during development becomes discontinuous over the course of development. And so we decided to ask this question and perform very simple labeling assays using lipophilic dye to essentially that is applied to the surface, either a PL surface or a ventricular surface, and look at the morphology of these cells over time. And when we perform this, experiment at early stages of development, either applying the dye crystals at the ventricular surface or at the PL surface, you get very nice labeling of the continuous radial glia fibers throughout development. But to our surprise, when we performed this experiment at later stages of development, the labeling of the radial glia fibers at the ventricular surface only labeled cells up until about the outer subventricular zone, And the converse experiment only labeled cells which terminated in cell bodies in the OSVZ, suggesting that indeed, during primate development, this transition from a continuous to a discontinuous radial glia scaffold is something that is quite different from the developmental histories of mice. And then what was very interesting was that when we looked more closely at these terminal branches of radial glia in the, emerging from the ventricular zone, we noticed something that was quite interesting in that many of these fibers actually fasciculated along the thalamocortical projections, and they sometimes ended up in those growth cone-like structures that we still don't understand what they do. What is very interesting is that when you look at the generation of neuronal subtypes and the timing of their generation, the switch that occurs during cortical development from deep cortical layer neuron generation, including markers such as TBR1 for the corticothalamic neurons and CTIP2 for the pyramidal tract neurons, it seems as though at the time when the radial glia scaffold transitions from the continuous to discontinuous structure, largely coincides with the switch from deep or infragranular to upper layer or supragranular layer neurogenesis in the human. And that led us to propose a sort of comparative model for cortical development mechanisms and the interactions between cell types in both mouse and the human and non-human primate model that accounts for the greater expansion of supragranular layers and has certain interesting implications that we can discuss later. We think that the discontinuity of the radial glia scaffold and apparently appearance of these radial glia fibers fasciculating on the thalamocortical axons could have implications for the patterns of neuronal migration, such that when the scaffold is more continuous, that supports more radial migration, versus when the scaffold becomes discontinuous and the cells have to sort of transition between different scaffolds, that may lead to greater tangential expansions of clonal-related cells during development. Next, as I said, most of these developmental processes seem to occur similarly across different areas. So how do you generate the regional diversity in the developing brain? As I mentioned earlier on, it's been appreciated since many decades ago that the cellular composition and the distribution and even the lamination of the cerebrocortical areas has been studied for many decades, and it's been appreciated. It's very diverse, and yet there seems to be this homology, this serial homology of developmental mechanisms. So how do you arrive at this diversity? It has been appreciated since several years ago that developmental gradients of transcription factors establish or may establish or lead to differences in the cellular compositions in different areas. And it has also been appreciated or hypothesized that expression of genes selectively in different areas of the cerebral cortex may lay out a protocortex that emergent axons or thalamocortical axons especially may selectively choose to terminate in different cortical areas. So in order to address this in a sort of more unbiased way using single-cell sequencing, we decided to collect or sample cells from several different areas of the emerging telencephalon or the emerging forebrain, and particularly focusing on three regions, two regions that are expected to produce very distinct cell types. So the cortical neuroepithelium that is expected to produce excitatory neurons, and then ventral telencephalic area, the medial ganglionic eminence, which in the humans and mice is the major source of cortical interneurons, where we expect to find the major differences between these cell types. And when we performed, in addition in the cortex, we also, for several of the specimens that we were able to acquire, we were actually able to identify the prefrontal cortex and the visual cortex based on the early anatomical distinctions that are found, and we were also able to capture cells from these two anatomically defined regions. And when we performed, again, unbiased single-cell RNA sequencing and analysis, we can identify the major lineages of the developing cortex, including the excitatory neurons, including radial glia cells, intermediate progenitor cells, and then newborn excitatory neurons and maturing excitatory neurons, and then the ventral encephalic or inhibitory neuron lineage that is distinguished over here and labeled with distinct markers. Now, when we overlay the anatomical area of information, things start to seem very interesting, and especially when you look at the comparison between cells that were sampled from the prefrontal cortex versus the visual cortex. When you look at the different populations, you quickly appreciate that both the radial glia cells, interneurons, and newborn excitatory neurons tend to intermingle such that these transcriptomic states are conserved across areas. But as soon as you start looking more closely at the maturing excitatory neurons, and this is all at the stages of neurogenesis, you already appreciate that different clusters are actually contain predominance of cells sampled from a single area, suggesting that the development of area-specific excitatory neurons that you've heard about from Bosilka's talk earlier today may actually be already apparent at these very early stages of development before sensory experience. So these cells are already born very early on development that are quite different. Using this data set, we were, of course, able to identify molecular markers that are both layer or cell type and area specific. For example, CPNE8, which is a marker of upper layer prefrontal cortex neurons, and hypocretin receptor 2, a GPCR coupled receptor that is a receptor for orexin, which seems to be selectively expressed in layer 6B or the subplate of the cortex. We wanted to also try to take a step further and start asking the questions, well, does it really matter that these cells develop such different or distinct transcriptomic states? Could it have implications, for example, for cortical connectivity? And in developing nervous system, it is quite difficult to study connectivity and the relationship or the correlates of connectivity from transcriptomic states. But nonetheless, studies in mouse have identified several interesting candidates, including these two genes, SADB2, which is a marker of intratelencephalic or callosal projection neurons, and BCL11B, that is a marker of pyramidal tract neurons. And what was very surprising was when we projected the expression data on our map, we noted that in the visual cortex, the expression of these markers is selectively excluded from each other, whereas in the prefrontal cortex, something that was quite surprising to us, a very high proportion of cells co-express these markers. And given that these transcription factors regulate cortical connectivity, we sort of hypothesize that perhaps they could lead to differences in the emerging connectivity in the cortex. We were able to validate these differences in expression pattern in primary tissues. Again, looking at the prefrontal cortex, these markers are co-expressed. There are beautiful antibodies that are available. Whereas in the visual cortex, these are exclusively expressed independently. Now, as I mentioned earlier, during development, we know a lot about gradients. And these gradients are induced by signaling centers in different parts of the emerging telencephalon. And the transcription factors establish graded expression across the prospective cortical protomap. If we looked, for example, in different anatomical area of the cortex, would we see something that is more of a variation on a theme, or would we see sharp boundary, for example, that would that could lead to emergence of boundaries between functional areas? And so to this end, we leveraged the access to postmortem sections of the human brain and stained them for these two markers. And surprisingly, pretty much everywhere we looked, in the frontal areas of the cerebral cortex, we found these markers to be co-expressed, versus in the caudal or occipital areas, also in parietal, we see that the expression of these markers is restricted. We were further able to narrow down this area to a very, very narrow transition zone, somewhere around the central part of the developing brain, where within a span of about 500 micrometers, you really change or you switch from this pattern of expression to this pattern of expression. So we really sort of are thinking about sort of changing our perspective on the homology of cortical area formation and how homologous the developmental programs are across the prospective areas, and starting to think about potential implementation of the sort of proto-map or proto-region ideas where certain parts of the emerging cortical columns might be adopting one type of excitatory neuron types and perhaps also establishing different patterns of microcircuit connectivity, whereas other areas such as the sensory areas and especially the visual cortex may be generating very different cell types. And there could be many different levels of differentiation at which further distinctions in molecular identities are established. There could be differences at the level of radial glia neural stem cells that are established by the emerging protomap, and there could be further refinements by the postmitotic or postmitotic phase specification imposed by the emerging thalamocortical activity. But this is currently a model that we're very, very interested in exploring, and how that translates into mature circuits in the adult is something that we would love to be able to explore with some of the emerging technologies that allow us to look at that. For that, of course, we need more suitable models that allow us to sort of pursue these questions and how do you model human neural circuits, cortical microcircuits in a scalable way in a culture system. So many of you have heard there's a lot of interest now in differentiating human pluripotent stem cells into cerebral organoids, and these structures leverage the properties of self-assembly of the tissue. The neural stem cell-like cells especially seem to recapitulate some of the same properties of self-assembly during development, and they generate postmitotic neurons. But there is quite a lot of interest in trying to understand to what extent these in vitro correlates of normal development recapitulate some of the same cell types that you may expect to find in vivo. And so towards this end, we've been, again, in collaboration with Alex Pollan and Aparna Baduri, we've been looking at trying to answer this question using single-cell RNA sequencing from across many individuals, many lines, and many organoids at different stages of development, and trying to compare this really to the primary cells. And one thing that is immediately apparent is that when you perform some of this co-clustering, again, a technique that allows you to merge data sets even though they may be generated using slightly different technology, or maybe your sequencing depth is slightly different. But overall, what we find is that you indeed can assign cells from the cerebral organoid to their in vivo counterpart, and that seems to be, and we seem to be able to actually identify homologous cell types across models. When you compare gene expression at the sort of unbiased cell type specific manner, you actually start to see some of the differences, and especially over here is a scatter plot where you compare gene expression level within a radial glia class across cerebral organoid and primary cells. And as you can see, red here are shown differentially expressed genes. And as you may appreciate, there's an overall quite a good correlation in some of the best markers of radial glia in primary and in vitro derived cells are very, very related. But then there are quite a lot of differences that we think may be important to the fidelity of these models. As you may appreciate, it is quite difficult to recapitulate the entirety of cell diversity in an organoid model. For example, some of the monocyte populations like microglia don't spontaneously emerge using most protocols that we've employed, and therefore what we can and are doing is we're actually deriving these cells in vitro using different protocols, and we've established protocols to label these cells and to transplant them into an organoid such that you can start to reconstitute some of the cellular diversity and start using interesting technologies for studying their intercellular interactions. We're collaborating with the Chan Zuckerberg Biohub to start employing machine learning on the dynamic videos that we were able to acquire to start learning about how these cells interact with each other. And I think that will be a very useful model going forward for studying various developmental processes. We also asked the question whether we can use some of the markers identified from normal developing brain tissue for regional identity, and is the regional identity recapitulated in an organoid model? So we, by taking some of these genes differentially expressed between different areas, we can actually use that as a classifier to assign a regional identity of excitatory neurons derived in an organoid. And as you can see over here, the prefrontal cortex excitatory neurons are quite assigned. These are primary. The classifier works very well and quite well for the visual cortex. But when you apply this classifier to study the heterogeneity or regional identity in an organoid excitatory neurons, you see quite a lot of variation. And actually, a lot of this variation comes from line-to-line variation. So some of the organoids, even though they've been exposed to the same developmental signaling pathways and they've been induced and differentiated using the same protocol, they actually acquire quite different, quite variable states. Some organoids tend to have the probability to differentiate into visual cortex like neurons. Some other organoid lines are actually have a propensity to generate prefrontal excitatory neurons. And we think that going forward, we will need to learn more about the fidelity and the reproducibility of these organoid models, and how can you use some of the lessons from normal development to guide the differentiation protocols to generate organoids that will resemble more of the prefrontal cortex or visual cortex, or also expanding this to other cortical areas. And we think that this will be a very, very useful model going forward. So to summarize my talk, I talked about how we employed single-cell analysis to try and identify and characterize the diversity of radial glia subtypes in the developing brain, and how that helped us identify some of the differences between human and mouse cortical development. We looked at the heterogeneity of differentiation programs, and especially the early evidence for very early emergence of area-specific excitatory neurons that we were very excited about. And how we are now trying to recapitulate some of the same processes in vitro using cerebral organoids. And I would like to thank people who were most involved, in particular Alex Pollan and Aparna Baduri. Both Alex and I were lucky to establish our labs at UCSF, and we'd love to acknowledge the funding, especially NIH and Chan Zuckerberg initiative. And thank you all for listening, and happy to take questions.
Thanks, Tom. That was really exciting. We have time for a few questions. Does anyone?
Very interesting talk. So my question is, you showed these interesting differences between V1 and the frontal cortex of SAPI2 and BCL11B, which we also observed in our adult mouse, of course. But do you see the corresponding differences in progenitors as well?
That's an excellent question. So the question is, where in the lineage differentiation process do you start seeing differences between areas? And the answer, there is no easy answer. When you compare gene expression between cells of the same type sampled from different regions, you will almost always find differences. The pretty much only difference is interneurons, which seem to be the most similar. But when you look at the number of gene expressed, differentially expressed, you see differences in radial glia, intermediate progenitor cells, but then the most differences are in the excitatory neurons, and that's where you start seeing actual cell type specific or area specific clusters. What we, however, think or would argue is some of the differences that are present and found already at the level of neural stem cells include even some transcription factors and genes that may regulate or initiate a cascade, transcriptional cascade, that would potentially then become amplified over the course of differentiation and essentially lead to exaggeration of these transcriptional differences that are area specific as over the course of differentiation.
Is it possible that these clusters that you see in different areas are homologous? In other words, that they represent as different just like with species, no, that they sort of evolve into a cohort of genes that essentially make this same cell type, but with different elements?
Yes, that's another excellent question. So can we identify, for example, neurons that are prospective or we think may eventually end up in layer 6 across different areas? And the answer is yes. We can identify cells that, for example, express TBR1, a transcription factor that is believed to be sort of driving the specification of layer 6 neurons. The same is true for the pyramidal tract neurons. We can identify cells that highly express FESF2. And the two clusters that we probably have the most, are least certain about, they express SADB2 and some of the upper layer markers. So some of the early distinctions between lamina may be present. And so indeed, we may be able to find some homology at the level of layer identity. So we can indeed, but for some reason, the transcriptomic differences are so robust that they actually lead to these clusters splitting differently. Now, that may change as we start to sample more regions, but so far, sampling more cells only revealed the same pattern. So sampling the larger number of cells using 10x technology, 10x genomics from these two different areas led us to very similar observations that the radial glia and progenitor cells tend to co-cluster. They still have differences in gene expression, including transcription factors, but it's the excitatory neurons that actually just seem to generate distinct clusters.
Cool. If there are no more questions, we can, thanks, Tom.
Thank you.
Again.